Greetings to all of our friends of astrobiology, and welcome to Ask an Astrobiologist, the show that celebrates the science and the scientists of astrobiology. We're brought to you by SegaNet.org and the NASA Astrobiology program. I'm Dr. Graham Lau, your co-host this month. Normally you see Sanjoy-san, but now you get me. I'm very excited for today's episode. We have a wonderful guest in-house. But first, we have a few little things to get through for our show, as usual. First off is our background quiz that we do every single month. You might notice right now behind me is a pretty cool looking picture of some place here on the planet that has some astrobiological relevancy to our search for extraterrestrial life and the quest to better understand the nature of life in the cosmos. Well, last month's picture that was behind Sanjoy, which you're going to see now on your screen, is a picture from Turkmenistan. And we put this quiz out for people to answer, and this month we had a winner who is Tejal Akaria, and I apologize if I'm pronouncing that wrong, Tejal. My pronunciation isn't always the best, but our winner, who's selected at random from all of those who have the right answer, gets some of our NASA stickers and some of the graphic history comic books from NASA Astrobiology. Now, the cool thing about that Darvaza gas crater in that picture is that it's actually not a natural site. This is a site that had a lot of methane or natural gas coming out of it back in the late 1960s, early 1970s. And some scientists who were working there, some geoscientists, wanted to look for natural gas and happened to have this crater collapse right below where they were doing some research. Well, they saw all this methane coming out and decided to set it on fire, thinking that would burn the natural gas away. Well, that was in 1971, we think, when it first was lit, and it's been burning continuously since then. Now, so far, we know of one person who's gone down into the crater to sample for microbes to see what kind of things are living down there. And this kind of research can be important for understanding organisms that live in not just hot environments, but environments with burning materials around them and lots of natural gas. So congratulations to Tejal for winning our contest for this month. We also have our Ambassador of the Month to announce again, and this month is actually a two-timer. So user AstroBioKash, or Kashish Nath, has won yet again. He tweeted, I think, 5 or 6 times about our show going into this month. So Kashish, thank you very much for all your support. Thank you to our dedicated audience who watch our show, who tune in and ask questions, promote us through Twitter and Facebook and Instagram and LinkedIn and all over the interwebs. We really, really appreciate that. So that's all the fun stuff. Now we can actually get to the real meat of our episode. Today's guest is Dr. Bruce Damer, who's actually joining me right now from NASA Astrobiology Institute headquarters at the NASA Ames Research Center. So Dr. Damer, hi, and welcome to Ask an Astrobiologist.
Hello, Graham.
It's great to have you here. I have to admit, in getting ready for today's episode, I listened to episodes of your podcast, Levity Zone. I listened to other episodes you've been in. I watched videos and read articles about you, and you've done so many amazing things in studying the origin of life and using computers to simulate ideas about how life can originate and evolve. You've worked on spacecraft design, so many things. But I think for our audience, we should jump back just a little bit, and I just wonder if you could give our audience a little bit of an idea of who you are and where you've come from and what things drove you to become a scientist.
Yeah, I was a rambunctious, dreamy teenager in Canada, in Kamloops, British Columbia, Canada. And when I was 14, I had a flash inspiration to work on the origin of life. I thought the self-assembly of molecules like Tinker Toy or Meccano, which we all played with, was a strange and interesting problem. And so I started kind of doing thought experiments, and I wrote programs, and then I met David Damer at UCSC, and I had developed a complete model by that point, and then we placed the model into the chemistry and then into the geological environment, and the model's working now, testing in the lab and out in the field. It's an amazing thing from a 14-year-old all the way to getting something that actually tests.
Yeah, that's absolutely incredible to have that kind of ambition at 14 years old that really kind of drove you on a career path, because you studied computer science, I believe, originally, and then eventually came back to pursue your PhD. Is that correct?
Yeah, I did computer science, and it was too soon. I literally, in 1985, wanted to use computers to simulate an origin of life or emergent phenomena with cellular automata. And then I discovered in 1985 it was too soon. I was like Back to the Future. I was like Marty McFly. I needed to go into the future. So 22 years later, I finished my PhD in this very topic, which provided the statistical basis for this new hot spring origin scenario.
Wow, that's incredible. So let's talk about the scenario then, this hypothesis that you've developed with Dave Deemer at UCSC. So what is this new hypothesis for the origin of life, and why is this a very strong model, you think, for how it might have started right here on Earth?
Well, until recently, a lot of science has focused on hydrothermal vents deep in the oceans as a place where life might start. But it was only a proposal. It's never worked actually in testing, i.e., if you define an origin of life as the formation of long enough polymers to then either fold or serve as data stores for information, and that those polymers have to be selected in vast numbers to be able to pull what's called molecular evolution forward toward life. You just can't do it in the hydrothermal vent in the ocean because water simply breaks everything up, and you can't form membranous compartments in that environment made of lipids. Whereas coming from space, for example, here's a ground up powder of the Murray meteorite, which fell on Earth, which was similar to the material that was falling on Earth 4.2 billion years ago. And if I smell it, I can smell organic compounds, amino acids, in this case, polycyclic aromatic compounds, nucleobases, fatty acids, the building blocks of membranes. And this material was falling on the land. And if it fell in the ocean, it would be too dilute. So if it falls on land, it goes into little ponds. And the little ponds can concentrate it enough to form the vesicular protocells. And then what Dave Deemer had discovered is that if those ponds undergo wet-dry cycling with the filling of hot springs or dew or condensation, but basically hot springs give you that regularity, then you can actually grow polymers. The polymers will form naturally without enzymes in between layers of the lipid that form a kind of bathtub ring slurry on the bottom. And so the wet-dry cycling that was featured, this is our Scientific American article from a couple of years ago, it's a cycle that simply goes around wet and dry, wet and dry. And through that, you have an engine, sort of a genesis engine, that can pull molecular functions or polymers to function. And then you get these little things called protocells that have increasing functional utility that make them more robust. You get more of them, you get a bigger sludge, and that sludge forms a unit called the progenote, which is basically, we propose, the common ancestor of all microbial communities. And we're testing this now in the field in Yellowstone National Park, in Rotorua, in New Zealand, forming these protocells from this prebiotic meteoritic material, basically simulants of it. And it's we're growing very long polymers of RNA, hundreds of bases long, and now increasingly other groups are doing peptides and deoxypeptides and other polymers you would need to start life through wet-dry cycling in a surface pool.
That's so incredible. I know in the geological sciences, there is some debate currently about when continents first formed and when there would have been dry landmass on the early Earth. So for this model, it really does require that there were continents very, very early on. If those continents really were there, where do you think that material ended up becoming in our modern world? Where would those old rocks have gone?
Well, the interesting thing is, if you look at post-planet formation when the oceans formed, there would have been a huge amount of cooling of the crust of the mantle, forming basically large volcanic protocontinents. So prior to the existence of plate tectonics or actual true continents, you would have had a lot of volcanic landmasses, which is ideal, similar to, say, New Zealand. It's ideal for the formation of these hot spring environments. And in fact, the entire Earth was a huge hydrothermal system with a lot of hot water going through rock everywhere under the oceans, in these volcanic land masses. So there's copious dry land that we predict would have been there due to just the simple amount of volcanism.
That's awesome. Do you think then with this model that there could have also been multiple forms of origins kind of happening in these different places with these cycles of wetting and drying, kind of bringing around all these different progenites that kind of came together then and really formed the first material? Or do you think it all happened in just one place?
That's a great question. And in fact, what we think is just like life today, life gets started in local environments and then can go extinct. But as long as it can distribute seeds or spores, it can distribute itself, it avoids going extinct altogether. So even in the Progenian, in this period we're calling the Progenian, where it's the boot-up phase of life, the protocellular masses, the fact that they're transportable, either being washing from one pool to the next or blown as a dry film, which is a way microbial communities still distribute today, they have a coverage across the landscape. So all the eggs aren't in one basket. And we think that that's the mechanism for the protogenomic distribution, in a sense, the horizontal gene transfer of all those innovations across the landscape, so that when one area becomes inviable, things go extinct. I mean, they're not alive yet. They just simply stop working. Other pools and other areas can support the continued evolution. So you have this grid, you have a network effect.
No, that's really cool. I mean, it's incredible research, and it feels like we're at a really important time right now in astrobiology, but also for humanity, where we're learning more and more about how it might have actually happened here on Earth. And we're also learning more and more about where else it might have happened in our solar system or even beyond, now that we know of thousands of exoplanets. I'm wondering if you have any worlds in our solar system you think are very likely candidates for life outside of Earth, or other worlds you think are likely outside of our solar system.
Primarily, we're looking at Mars, because the amazing thing about Mars is the Spirit and Opportunity mission, the MER missions. Spirit, driving toward the end of its mission, had basically one of its wheels stop turning. And they actually then use it as a trenching tool, and they trenched into the area at Columbia Hills they were driving through, turned up a white powder. That was opaline silica. So they realized we have happened upon, by accident, an old Yellowstone type environment, preserved 3.7 billion years old, just as old as the stromatolite environments that we find these in Australia and Western Australia. And so suddenly they realized all these rocks around us are maybe these digitate silica nodules that might contain evidence for life, because we know that old hot springs on Earth always have life, and they always preserve evidence for life very well. So we think that Columbia Hills, where Spirit is a good place for returning to Mars and doing a sample return mission. On the other hand, our colleagues that are looking at the plumes coming off through the ice sheets, the ice crust of Enceladus, environments like Titan, may not be so conducive because they don't have wet-dry cycling. And in fact, the energy available in the ocean, even if there is a hydrothermal activity, the energy flux is so low in those environments without access to sunlight, and with the coldness and the limited scale that there might be hydrothermal activity, that life just can't get started in an icy ocean world.
Yeah, so it sounds like Mars could have then been a good target, especially if life could start in some of these siliceous center kind of areas. I wonder what you think about Venus then, long ago in the early solar system. Obviously, we don't know right now what early Venus was exactly like, but even last week, I think at the DPS meeting in Europe, another paper came out talking about the potential for Venus being habitable for life as we know it in its early history, maybe for a long period of time. Do you think there could have been life on Venus, especially with this wetting, drying, cyclical structure in hydrothermal environments or geothermal environments long ago?
Well, certainly, and I saw that article too, and certainly there could have, and it's a chilling tale, or literally a heating tale for us, that Venus's oceans, it was essentially went out of habitability, and the oceans evaporated into the atmosphere, and then the hydrogen was stripped off, leaving a CO2 90 bar atmosphere. If life started on Venus, just as in Mars, if life started there, then perhaps the only place it could survive is seeking refuge in the rocks. So hot, wet rocks, and it's difficult enough to drill on Mars to find evidence of active microbial activity. We see some methane release, but Venus, they would have to be surely in the rocks. The chilling part of all this is that planets don't necessarily maintain liquid water for 4 billion years. It's not a given. In fact, it may be a very rare case, and it may be that Earth just got extremely lucky, maybe perhaps with the moon that we have, the exact size of that moon, or some factor kept liquid water on our surface long enough for complex life to rise. So it is truly a chilling tale that we may be extraordinarily rare, not for microbial life to arise, but for complex life to arise.
Interesting. That kind of reminds me of the rare Earth hypothesis from Peter Ward and Don Brownlee some years back, considering all these various factors that we have to think about with our Earth and our position right now in the cosmos, and how rare worlds like ours might be, not for life as you mentioned, but for more advanced life to form, more intellectual life that can look back on itself and realize it is alive. And there's so many cool things we can keep talking about on origin of life stuff. I do want to jump a little bit now. I know that you've had some background in developing spacecraft, and more recently in working on spacecraft for harvesting resources from asteroids. I wonder if you can talk a bit about that work that you've done and what you think the future holds for sending spacecraft out to do some of this work of harvesting resources.
Yeah, it was about 6 or 7 years ago that NASA had the Asteroid Redirect program. People may have remembered that. Our team, Peter Janiskins and Julian Nutt, Peter's from SETI Institute, Julian's an independent balloon designer, we came up with this idea that if you came up to the asteroid, matched its orbit, extended a regular helium balloon structure that you sealed the end of, introduced a controlling gas, you would actually stop the tumbling of the asteroid, and you would be able to direct it with a flow of gas and put delta V, which is the changing of orbit, and then drive along as a coupled unit without touching the surface. You can't touch these objects, you can't wrap cables around them and mine them as people have proposed in the past. They're potentially loosely consolidated rubble piles. But then we could literally move asteroids, so we could get to lunar orbit, release one for geological sampling for people like you who would love to go with a rock hammer and sample it with a backpack. But also then we realized we could extract volatiles. Water would come off and condense into tanks, and we could turn that into fueling stations. We could also use the Mond process of electroforming to extract nickel and iron with an electric field and plate large objects in space to build these big space habitats that Jeff Bezos is talking about, the O'Neill colonies. And the last thing, we could turn a 50% water ice, potentially with carbonaceous asteroid, into a globule that would support a biosphere to feed people in space. So the three of us realized that this Shepherd idea, we call it Shepherd, could be the single technology, and in fact like a sailing ship in space, a single technology to open the solar system and do incredible science.
That's awesome. Yeah, and when I was first posting some stuff to try to get people interested in this episode, I mentioned spacecraft design and asked some people what their favorite spacecraft were from science fiction and from reality. And we had NASA respond back with loving the SLS, the upcoming mission rocket to take us with Artemis back to the moon. There were some other responses, including from me. For instance, I love the idea of organic being ships, or ships that are like slightly organic. So for instance, one of my favorite episodes of Star Trek The Next Generation had a character named Gomtu, who himself was a spaceship and who could interact with his crew. And so I wonder, from your side of it, what are your favorite spacecraft from science fiction or from reality, and why?
I really liked Wernher von Braun's and Chesley Bonestel's descriptions in the 1950s and the early 50s, which really launched the space age. You know, they had the rotating colony, they designed spacesuits, they had winged spacecraft, they had gum-shaped landers. They really nailed it, right? And it's an amazing thing. But what they missed were fabric enclosures. So my favorites are, of course, Shepard's a fabric spacecraft, but I liked Echo 1, the first telecommunications satellite in the 60s, I love Bob Bigelow's work in the A330 and the beam on station. It's just breathtakingly interesting work because suddenly you can launch an entire station on a single lift. And to go to Mars, you have an envelope, a couple of meters with the water that you get from asteroids, and you have the radiation shielding problem solved. And so I think inflatables, as they call them, are the future.
That's awesome. Yeah. I think a lot of us would love that to be the future for us to go into low Earth orbit and just have a chance to see the Earth from space, honestly. So before we open it up to questions, and just a reminder for our audience, those of you watching on SegaNet and on the NASA Astrobiology Facebook, you can ask questions in the chat there. You can also ask us questions on Twitter using hashtag AskAstrobio. And if you're watching this after the fact on YouTube, feel free to leave some questions on YouTube as well. I come through and I answer those. And if I can, I'll also shoot them along to Bruce and our other guests who are in the YouTube videos as well. So before we open up the questions, I want to chat a bit then, Dr. Damer, about your work as a storyteller. You've done a lot in going out to the community, to the public, and speaking with people of various backgrounds about science and about the origin of life and about your research and telling poetry and giving talks at Burning Man. And I wonder if you could just speak to us about what really resonates for you and makes you feel the drive to go share your science in that way.
What resonates for me is, of course, that the next generation, you might call them millennials or iGens, people arbitrarily name generations, they are our future. So if they are passionately excited about science down to their core, like I was when I was 14, think almost as a spiritual quest, not just sort of a career choice, that I was completely to me, science is the most amazing story that humans have ever told, that we're unfolding the story of truth, the story of measurable and testable reality, and that the cosmos and the birth of complex life on Earth is the most awe-inspiring, mind-blowing story that's ever occurred. And so taking young people back into that progenian period, back to that period where you have fragile protocells that don't have a ribosome, that don't have a nucleus, that have bare bones metabolic processes, and you put them into that soup. You say, these little fellows are the ones that carried us all the way to complex life, and watching how quickly they fail, there's almost an emotional connection with that world of, my gosh, this is how improbable we are. This is the process where it started passively and then it became active. And the mind-blowingness of how on earth did this happen, but it did. And we are here and we're the beneficiaries of that. For me, that's a spiritual quest. And so if I can bring that to audiences like at Burning Man or the festivals, everyone's dressed up in a funny outfit. We've got live painting of artists like Android Jones going. We're reaching them in the medium that they care about, that it's the most powerful storytelling medium for science that we think that we can create for this next generation. And then they get excited. They join astrobiology groups. One of the young people in festival joined our lab and did fundamentally important work. They just get excited. So they don't get led astray by flat earths and things like this, which are a little dangerous to our society. You know, we risk losing science. We risk losing all this progress if people can't discern between made up stuff and conspiracy theories and this quest for truth. And I think they get that, but you have to put that out in front of them and say, this is why this is the most exciting story and thing to work on that we know of.
Oh, that's incredible. That really inspires me just hearing that. As a scientist and science communicator myself, and I think I was one of those young people who was impressionable and loved hearing these tales from people who cared to share them. So that's wonderful. One more thing before we open it up to the audience questions. I promise I'll get there soon. So Google and the SETI Institute have worked with NASA with the Frontier Development Lab, something called FDL. I know you've spoken there before about the future of artificial intelligence, of machine learning, and where computing will take us. I wonder if you can speak just a little bit about the future of astrobiology and maybe even the future of science right now through artificial intelligence, through machine learning, and what you see coming down the pipeline here.
I think that AI and machine learning are the greatest tools we have for this century, from everything from telemedicine to self-driving cars to figuring out solutions for climate change shocks that are coming. And after my presentation at FDL, where I showed our origin model, but when you abstract the origin model, it comes out as three parts: probability shaping, interconnection or interactivity, and memory writing and reading. We just abstracted our chemistry of protocells to those three things. And then it suddenly occurred to me one night, that could be the general principle of emergent systems, which is what I've been after since I was a teenager. And so the fellow from Google put his hand up and said, that is fascinating. We want to be involved. So we've been having meetings with them about how to ingest or inject biology and genes and evolution into AI itself. Because right now, AI is just a great big neural nets that you have to train and train and train manually. What if it could evolve and learn on its own through genetics and through the principles of life's origin? That we could have a truly powerful new generation of AI for the tools that we need in this century.
Yeah, that's awesome. So we will open it up to our audience now. Once again, SeganNet, NASA Astrobiology Facebook, please ask us questions. We'll get them into the queue here for Dr. Damer. If you're using Twitter, feel free to hit us with hashtag AskAstrobio. Our first question actually comes from one of our longtime fans of the show, Marianne Denton. Marianne asks, and she has a term here that I absolutely love. She says, if the wet, dry, and warm little ponds hypothesis is necessary for the development of polymers, or, as she phrases it, life's Legos, is it possible that those long-chain molecules could have migrated via weathering or volcanism to two hydrothermal vents in the ocean for finishing? And then her real question here is, does it have to be one or the other? Could it be some mixture of both?
We've had a lot of conversations with our colleagues on this, including the latest Astrobiology Science Conference in Washington. And we've really determined that it is one or the other. If you have a polymer that ends up in a liquid environment, like an ocean environment, it's going to break down due to hydrolytic degradation pretty quickly. So you really have to have access to surficial environments and the atmosphere to make the chemistry work. So in the oceans, you have just a mineral interface with rock and a water interface. On the surface, you have, mineral water, mineral air, water. It's just that much more complex and rich in environment. And you have many more pHs, rock weathering products, and access to that important exogenous meteoritic material. So that the difference between the surficial environment and the oceanic environment is profound. They really are two regimes. So what we think is that as protocells became living cells and living microbial communities, those communities would adapt to salty ocean water later down in the estuaries. They would flow downhill and have to develop the active pores to equilibrate all that salt out of the cells, which cells do today. And then only many, maybe hundreds of millions of years later, after adapting to the marine shoreline, you may find extremophile organisms adapting to hydrothermal vents in the submarine environment, which are truly extreme environments. So that's the path that we believe life took.
Interesting, yeah. So starting on land, then kind of spreading out to these tidal zones, then down into the oceans, and then evolving later to come back up around again, right? That's really intriguing. So our next question, I actually want to preface first. I heard you speaking to Michael Phillip in a podcast from some years ago, and I heard you mention in the podcast that you don't really buy into the ideas of the technological singularity and technological dystopias and some of these ideas from cyberpunk that kind of are in our culture now. So Gene Griffith on SeganNet wants to know which of the great filters you think might most be likely to stop intelligent life from being everywhere. And if it could be that the power of an individual or our technology really could be really what drives the end of a civilization.
I think the filter, because the filter theory, there's so many that prevent intelligent life. One of them being, which is crazy to think about it, according to primate biologists, apes, primates were going extinct at the time when Africa split down the middle and put our ancestors into the dry savanna. So apes were going extinct because monkeys were much better foragers, even though they had smaller brains, but they worked as a communal unit. And so our ancestors were going out anyway and probably wouldn't have survived. So the big brain thing that apes had wasn't a selective advantage in the end, potentially. But we were just cast out of Eden into this drying savanna, and that the monkeys weren't there. And then we had to adapt quickly. And in a sense, we are creatures of climate change. One of the things that occurred to me last week is that as these climate change shocks are coming, we were made by climate change. If you look throughout the evolution, when humans walked out of Africa, they walked straight into advancing glaciers in Europe and Asia. So we perhaps are evolved by climate shocks. So we may be pre-adapted to deal with this global climate change shocks that may be coming. But the second part of the question was, how do we go forward? I think we have to go forward by the same way that life rose. We have to link together into strong community again. We can't allow ourselves to be isolated and separated by our own processes and dialogue. It's really together we will get through this thing. So we can't afford this sort of stories of anxiety and separation. We have to be very positive, very gearheady, and go ahead and build our solutions for the future as a community, as a global community.
That's really incredible. It's weird to think of us as being driven by climate change to actually be able to adapt to climate change very quickly. We are certainly now children of the Anthropocene, so it's interesting to think about what a global consciousness, what global awareness could really do for us moving forward. Our next question comes to us from Twitter, from our ambassador of the month, Kashish, has asked the question. He's asked, according to theories, Mars and Earth formed pretty much around the same time. And if it's true that Mars has fossil life on it from back then, what could have made the life on Mars develop so fast and then also end so fast?
So if Mars had similar conditions in the Noachian period into the Hesperian period, where it had oceans, shallow seas, but it had precipitation and hydrological cycling, it would have had a lot of hot springs as well because it had volcanic activity. But unfortunately for Mars, it did not have a big enough protective magnetic field, so the atmosphere got stripped away rather quickly. Our colleagues at SETI think that maybe Mars was out of habitability at the surface by 400 million years into its history. Others say by 1 billion years, but certainly surface habitability was lost. So that's actually another argument for it. It's not a good idea to try to settle Mars because it's definitely a sterilizing sink environment. But life, if it started there, could have started rapidly. We just don't know. That's sort of an inference that now that we know that hydrothermal pools can cycle systems and that polymers have to grow at a certain rate and be resynthesized at a certain rate to stay in that kinetic trap, that we could say, well, then life must start quickly. However, that's an assumption. It may be that there was many starts and failures of the system before it found a combinatorial way through to robust, say, phototrophic cells that were finally more autonomous. So life's start may not have been quickly, and it may not have been quick, rather, and it may be that to get to robust life, to the life that uses ribosomal translational machinery, that may be a very circuitous path. So is the life on Mars robust enough to use sunlight, to leave stromatolites, to leave a fossil record? Perhaps not. We just don't know. That this is a great area for thought experiments.
That's awesome. And it also seems like a great reason to keep going back and keep searching for some of these potential signs of past life with new robots. And I guess I'm with my own question out then. So you did mention with Gusev maybe sending a mission back there for a quick sample return, or Columbia Hills, and bringing some material back. If you had the ability with no budget limit to send a mission to Mars, where would you send it? What would you do? And why is that important? Well,
the amazing thing, sometimes nature throws you a ringer. It throws you a wonderful chance. And with Spirit coming into Columbia Hills at the end of its mission up to the home plate formation, then finishing the mission there, what it imaged were these little digitate rocks, these silica nodules all over the surface, plus the outcrop, plus the opaline silica that was trenched up. To get some of those nodules, they look like the nodules that the Maori call land coral in New Zealand. They look like the structures in El Tatio in Chile. So to actually return those just on their own would be of huge astrobiological value.
Awesome. Yeah, it seems like we have so many good reasons to go back and take more samples, but also to do sample return at some point soon. Our next question comes to us from another long-term fan of the show. Dr. Jim Pass is the president of the Astrosociology Research Institute. He's asked on Twitter, what social and cultural impacts does the search for extraterrestrial life produce here on Earth, even though we really haven't located yet any definitive proof that there is extraterrestrial life?
You know, Jim, that's a really good question. And I know you were just recently out in the Pilbara in Western Australia, and I hope you were able to bring some of this stromatolite home for your desk. But the search for a life has a huge impact, I think, on our species. For example, what if we can't find evidence for life outside the Earth, but we can do something secondary. We can set up science such that we can see the first progenotes in the laboratory. They're undergoing the first evolutionary steps toward life, not anywhere close to life, but we see pores and metabolic functions and stabilizing polymers be selected in molecular evolution, and the mass of progenotes grows in size. We hit it with some kind of a stress, it shrinks and crashes and then grows again. And that'll be the Turing test for whether something is going toward the living world. And I think the images of that happening in the lab will be as powerful as seeing Earth from space, from Apollo 8, those first wonderful pictures. And so it's something we could actually even anticipate or prepare for, that science could say, this is a plausible, we don't know and we never will know how life. This is a plausible argument for a mechanism for life to start on a world. And then it, of course, tunes our search in exoplanets for which worlds could support this. And it will be a very big, in a sense, spiritual impact or philosophical impact on our species. It'll be perhaps the first, if you will, alien that we make. But it's not that alien in the sense it's a laboratory evolved system. But for sure, it won't look like what the true progenotes were back 4.2 billion years ago. It'll be taking a different path, as Carl Sagan or Stephen Jay Gould's idea of rewinding the tape and then going forward, and you get a different version every time.
Mm-hmm. Yeah, it seems powerful, the idea, too, that we could be the creators of life in the laboratory while attempting to understand how we came to be in the first place. That seems like a very powerful moment for us as a species. And our next question, I think you kind of already answered, so I'm going to add to it a little bit. This is from GS Hurd on SaganNet. He asks, have you guys started a lab simulation as well as the work in Yellowstone? And I'd like to go a little bit further then to ask, what's actually going on in the laboratory that's allowing us to test these models? And what kind of time frame do you think that we're looking at scientifically for laboratory simulations to actually bring us to the point where we think we might have our minds around how it could happen?
This is a wonderful shout out to everyone out here, because I'm fundamentally trained as a computer scientist. I came into this hoping that computers would be able to do a simulation that would guide the chemistry. Found that the computers weren't fast enough where we didn't understand the chemistry. Jumped into the chemistry working with Dave Deemer and our colleagues. Getting the chemistry now to work, we can reverse the thing and make computer science models of these polymers, of these selection criteria. We've done it for the kinetic trap to show that that's real and characterize how polymers grow and continue to grow in length and continue to work in this kinetic trap away from equilibrium. What about protocells? Could somebody out there write a simulation of protocells, even low fidelity, that would show us the likelihood of the emergence of the first pore, for example, the first passive pore, and how that would affect the units. I think it's a great PhD project. It's a great at-home type project to simulate our laboratory and fieldwork conditions that are working, that are creating emergent phenomena. Because then the computing will get so good, and maybe you can throw it all into a huge cloud-based system that will guide future chemistry, and we can do it like this. We can stack up the efforts and accelerate them.
That's awesome. And I guess I also wonder then, if you had to give advice to anyone right now listening at home or watching our show or listening to the podcast version of it, who wants to be involved, if they're a high school student or a college student, or they're a professional working somewhere else, but want to be involved in the computer simulations or the lab simulations or the field work, what would your advice be to those people to get involved in this kind of work?
I think that become a multidisciplinarian, in that in the field of astrobiology, usually each person in the field has at least 2 disciplines, but maybe get 4 or 5. So study complexity science, study membrane biophysics, study organic chemistry, polymerization or polymers, study how cells work, then study some geology. Go out and sit next to these hot spring environments, and you know as a geologist that it's such a great teacher that you can see at the lake shore or next to a pond or something, you can see all this complexity that the natural environment provides. You can only do that by going to the field. Become fascinated in all those things, and you'll start having kind of visionary downloads of how they all connect. And then you might want to go the geology route or the chemistry route, but you may want to go the computing route or collaborate and create these multidisciplinary teams, and then build your own little laboratory instrument that is a rocker plate going back and forth, putting fluid over a mineral with the lipids and with the monomers of the polymers, and just build something at home and then scrape that off and do sequencing on that and see what you can see. And you could build your own experiment. And then effectively, Dave and I have sometimes done our experiments in test on frying pans on stovetops in the kitchen.
That's awesome. Kind of makes me think just weirdly about like biohacking, and how there's a lot of folks right now who are like taking their own materials back at home and trying to like learn how to hack through genes and genetically engineer cells. It'd be pretty cool if we had a bunch of people who are also trying to do origin of life hacking at home in that manner. That's a pretty awesome idea. We do have a bunch more questions. I promise to get to as many as I can here. Our next question is from Tom Caruso, who's a longtime fan of the show. Tom asks us on Facebook, is it possible for you to apply the wet-dry cycles to the internal cores of icy moons covered by deep oceans, such as Enceladus and Europa? The premise being that gas is produced from serpentine reactions and orbital oscillations, allowing a loosely bound rocky core to stretch and relax, could possibly produce the dry environment, and then mechanical driving force to alternate between wet and dry.
We've looked at that with our colleagues, and our colleagues in geochemistry have basically said that in any environment which is under the pressure of a water column, there is no way to have reduced water activity. So there'd be a gel, a temporary cavity. You're always going to have the presence of water, so you cannot do the condensation reactions that we're doing with wet-dry cycling. So then you might say, well, maybe within the ice that there's a pocket, that there's some air that is contained, so there could be some drying. The problem is the ice is going to be so cold, it's not going to have enough activation energy for the chemistry. And then if you had, say, a few polymers forming, there's nowhere for them to go. And you also need access to the membranous lipid material, which isn't going to be prebiotically plausible in that environment, to create the compartments to crowd everything enough to make the chemistry work. So if someone can build an environment in the lab that shows that this is possible in an icy and watery environment, all power to them. But if you just look at the thermodynamics, the barriers are just extremely high to any of that working in a watery or a cooler environment.
That's awesome. So that's a good answer. I wonder what Tom thinks about that. I'll have to talk with him later about that. Our next question comes from user Vern Emmerich on Twitter. Vern wants to know, since all of life on Earth appears to have evolved from a single common ancestor or a progenote, shouldn't that same dynamic be true for a galaxy? So in other words, once an organism achieves some level of homeostasis needed to traverse through sterile environments, traverse the galaxy, and then it starts to spread and evolve, would it become that organism kind of becomes that universal organism as well? It spreads faster than other organisms that might be arising in other places.
That's a very good hypothesis. It's sort of an extended panspermia hypothesis. So, for example, if we could catch up to an extraterrestrial, extrasolar object coming through the solar system, get it, sample it, find bugs on it effectively, sequence them, and see if it has the same genetic code as to ours on Earth, it would answer one of Chris McKay's big chart questions of where life can start and is it common origin. Did you need to have the hydrosphere of a galaxy to combinatorially start life? And then it just seeds through this rocky transfer, these desiccated, low metabolism transits. My approach is that that's a complicated solution, that perhaps life, since it's so exquisitely evolved hand in glove to the Earth environment, it probably did start here. It could have started on Mars and come through in a transit, deposited on the Earth, but it doesn't. It really pushes the question of where life's origins would occur back to some other location. And I don't think it changes really the nature of the questions. You still have to form long chain polymers in an aqueous environment. You still have to encapsulate them in a cell-sized compartment. As far as we know, that's the only way to get function to come out of matter.
Interesting. I wonder, toss my own question in there, if you've heard of Leo C. Chen's dark forest hypothesis from his science fiction works, and this idea that, say you're alone in the jungle at night and you want to know if other people are out there, you could call out into the forest, into the dark forest, but you might be attracting the leopards and jaguars and things that might eat you. And the idea is should we be sending messages out into space because we don't know who might be listening, even if we think that we might be looking for a friend out there. I wonder what you think about the potential for other extraterrestrial intelligence out there listening to us and whether they might be benevolent or malevolent.
I feel that complex intelligent life is probably so incredibly rare that if they got a signal, it would be as profound a philosophical impact on them as it would be for us. And they would be quite humble because they would have probably done this kind of thinking about their own origins, realized how difficult it is to get to intelligence, and been grateful that it occurred somewhere else. And certainly, we don't have much to offer. I don't think that the effort needed to mount an interstellar expedition to come to little old Earth and, pick up a burger at the local burger joint is going to satisfy. What they're going to be asked after is philosophical inquiry. What is our science? They're going to be very, very open. They're not going to be predating on us. There's nothing of value materially here, but our knowledge, our science, our arts are what they would be fascinated by.
Interesting. Yeah, trying to share culturally. That's awesome. Our next question is from Ramon Rocketeer on Twitter, which is an awesome username, by the way. Ramon asks, with a fragmented magnetic field on Mars, is one region marginally more protected than another from radiation effects for life?
I don't really have any idea. It's not my area.
Okay. No worries. We can skip past that one. Ramon, I'll reach out to you later and we can chat. James Mullins on SeganNet has asked, how can this theory for the origin of life help us develop our technology for terraforming new planets potentially? Which is an interesting question.
Yeah, I think that the concept of terraforming new planets is probably beyond us. I can't really imagine if you dropped a full comet onto Mars, its liquid water would simply be absorbed in the environment and continue to be lost. So Mars being the only candidate, I think I would dare say it's a pipe dream, I think, to terraform a place like that. I think that we can build biospheres, like with our Shepherd idea. The O'Neill colonies are much more viable, a kind of terraforming by building new terrestrial environments. Give you an example. If you took a cruise ship down to Antarctica, which you can, You might get off that cruise ship and go see Shackleton's hut and whatnot, but you'd be in a big hurry to get back to the cruise ship to have your hamburgers or your veggie burgers for lunch, because it's not habitable for humans to be in Antarctica without this extensive resupply and housing. And I think that's a metaphor for Mars. Mars even worse. The moon's even worse. So we're going to be building cruise ships. We're going to be building, hopefully not the Axiom, where we all get too big on milkshakes, but we're going to be building structures in space. I think that to try to terraform any surface is far too much effort and probably beyond our capacity.
Oh, interesting. I wonder what you think then about some ideas from science fiction that have happened in the past. And I know Carl Sagan wrote on this. Eric Vernquist made a beautiful film called Wanderers that has really great imaging of this in video. But the idea of us maybe taking asteroids and building spacecraft and habitats out of asteroids and hollowing them out and building our habitats inside of them, how feasible do you think that is for humanity maybe in the next 100 or 1,000 or 10,000 years?
I personally, I'm not an expert in asteroids, but have studied them through the work of Peter Janiskins. I think it's infeasible because mostly they're consolidated rubble piles. Their geotechnical properties, I don't think, would allow you to heat them or core them out. And you're dealing in a low gravity or virtually no gravity environment, so material's going everywhere. Very hard to operate in that environment with all this dangerous, basically large boulders moving around if they come loose. I think it's more likely that we're going to extract, perhaps with a gas, like we're talking about in Shepherd, pure metal ions, and then we'll plate them into the parts we need. Because asteroids are hazardous. They're hazardous to deal with. That's why in our design we don't touch them. We handle them gently with gas. They're not exactly a safe place to operate.
That's intriguing. So we have another question here from Bruno Pavletich from SaganNet. So Bruno, we talked earlier about this potential for life to start in the dry, wet cycles in these geothermal environments and then maybe migrate to intertidal regions and beyond. So Bruno wants to know how long you think these first progenites or these life forms should evolve before they can begin spreading out from that point of origin to then start inhabiting other places.
This is an excellent question that we'll never know precisely, but we can make some guesses. So, for example, at 3.5 billion years ago, 3.48 in this piece of hot string stromatolite discovered by Tara Jokic and Martin van Kranendonk, you can see clear evidence of a microbial ecosystem here in this geyserite, or on the layers on either side of the geyserite. So that's 3.48. There's some evidence that maybe at 3.7 we see stromatolites in Greenland. That's disputed. There's some chemical evidence that we might see life, evidence for life existing before 4 billion years ago. Then on the other side, oceans formed by perhaps about 4.3 billion years ago. So there's a 200 to 700 million year gap in there to get to, when you get to this, you have modern life. These leave the same telltale textures in the rock as you'll see in New Zealand, for example. They are tough. This is way past Luca, past the common ancestor. So it's hundreds of millions of years, perhaps, maybe tens of millions. I think from the progenian or the progenote to the microbial mat, we'll never have fossil evidence of that period. In the laboratory, we may be able to simulate the first few steps and make some guesses, but it's always going to be this guesswork about time frames.
Interesting. Yeah. I once told some geochemistry students that maybe the only way we'll ever know for sure is if there were intelligent aliens watching us from afar and they have some videos in a library somewhere for us to watch.
There is one other thing. If we find rocks on the moon from that period, right? If we find exogenous material on the lunar surface, or probably not Mars, but the moon, it would be great preservation of perhaps something containing evidence of stromatolites or microfossils.
Yeah, that's a really good point. I mean, it's been hypothesized that there could be, now extinct materials from early life on Earth just sitting on the moon for us to go find, which could be a good reason to go back.
Yeah.
Our next question is from Ben Pierce on Twitter. Hi, Ben. He says, do you think it's necessarily RNA-filled protocells that made up the first life? And then what do you think about other information polymers being first, perhaps outside of RNA, DNA? I know recently a paper came out suggesting an RNA-DNA hybrid. What about other potential genetic information systems?
Yeah, in fact, there's talk of there's many potential systems like origamis between RNA, nucleic acid-like polymers, and peptide. Possibly we had access to amino acids more so than the nucleotides that Ben works on at McMaster University. So could we have something that's a close cousin that still provides informational? There's been work on TNA, work with Ram Krishnamurti at Scripps, work at the Chemical Center for Chemical Evolution in Georgia Tech on, and other places on these non-canonical bases. Also in India, our colleagues at Pune in India. So yes, that is a field ripe for exploration, and we ought to extend our range of what we're using in our experiments to convince more of our colleagues that this is prebiotically plausible.
That's awesome. Our next question kind of brings us back to where we were a little earlier, considering potential or no potential for having life start in Europa or Enceladus's oceans. Preetha Jaipal on SeganNet has asked, if you think that this concentrated pool theory for the origin of life, or the wet-dry cycles, strictly pertain to conditions on early Earth, and the question then kind of comes down to, do you think that there's other ways that origins of life could occur in different kinds of environments on other places and other worlds in the universe?
It all comes down to definitions again. And of course, years ago, NASA held a conference to try to come up with a definition for life. What we're doing now in our field is coming up with what we call a crucial experiment. This is from John Platt's Ideas of Strong Inference from the 1960s, that we need to have a single sentence or a couple sentence long description of how life can begin that's abstract. And I've been recently talking to Laurie Barge at JPL and circulating this around all of our colleagues. And they're kind of coming to agreement that we need a non-enzymatic process to create polymers long enough that they have catalytic function, get them together in sets somehow so they can undergo large-scale combinatorial selection, which sets up molecular evolution. And you have to be able to keep the polymers around and resynthesize them. And I think that we're kind of coming to agreement that that's what's needed for an origin of life. So if you look at Titan, if you look at other environments, you have to be able to do that. So you can propose a chemistry that, as long as it meets that crucial experiment, it's a testable hypothesis. And so that's what we're putting out to our field, a new article coming out in Astrobiology in a few months.
Awesome. We'll look forward to it, and we'll share it through SeganNet's Twitter and Facebook, through Blue Marble Space, through National Astrobiology, through my own social media, of course. So we are running out of time now, so I want to say to all of our viewers who are watching right now on SeganNet.org, on the NASA Astrobiology Facebook page, and on the live stream, thanks so much for watching the show. If you had questions we didn't get to, I'm really sorry. We actually had the most questions ever for one of our shows for this episode. We didn't even get to nearly all of them here. There were just so many. So thank you to all of you at home who are watching, who asked all these wonderful questions. Dr. Damer, it's been great having you on the show. I do want to plug you a little bit here. You have damer.org is your website where people can go and read some more about
Damer.
you. Damer.org. And I mentioned earlier too that I've listened to the Levity Zone podcast. I've really enjoyed all the episodes I've listened to so far. They're really great. The first one on Genesis engines is incredible, and definitely worth a listen for our audience. And then, is there any last parting words you'd like to give our audience, our intrigued astrobiologists and astrobiology enthusiasts?
Yeah, I think that we are in an exciting, revolutionary time in the origin of life field, that because of the power of the field of astrobiology and all the minds that have come together and all the collaborations now, and that when we address the question of the origin of life, it's also our own origins, and that we hope that perhaps the discovery that we needed to have a communal unit at the beginning of life, not individuals in competition, but a community of simpler units that were in collaboration to start life, that you can actually see all of life through that lens. And that as we go into the future, it isn't competition between us as humans and us and the biosphere that is going to let us survive. That's a metaphor that's perhaps not constructive for our future. It is the collaborative communal unit that will give us our biggest chance of survival. And if we show that life itself came from that forests are made that way, that all ecosystems are made that way and thrive that way, we'll learn how to thrive through collaboration.
That's awesome. Well, thank you so much for that. And thank you so much for joining us on the show, Dr. Damer. It's been a pleasure having you here. I'm sure our audience really loved it, as shown by all the questions they had for you. I can try to share some more of those questions with you if you're interested after the show. To all of our audience members, we tried something new this month with our new graphics for the show. You'll see some more of the artwork from Aaron Gronstal from the Astrobiology graphic histories are in the show now. And we have some new things. And so we'd love to know what you guys think about what we're doing here through Twitter, through our YouTube. Let us know what you think. Our next episode will have the final versions of this new format for the show moving forward. I'm glad we also mentioned Lori Barge, because she actually is our next guest on the show as well. So expect that coming soon. Dr. Damer, to all of our guests, thank you very much for joining us. And remember, stay curious.